Okay. Uh, right. So we can continue fertilization in mammals. The last class we have looked, there are many sperm cells. It's millions of sperm cells, like 500 million sperm cells. When one sperm cell first arrive to contact with the ovum, then we have seen how these uh, membranes fused, an heterosome reaction enter the sperm nucleus, and immediately cortical granules make the zona pellucida harder. So then other sperm cannot enter, only one sperm cell can enter into the ovum. So this is called prevent polyspermy. Cortical reactions are important to prevent polyspermy. Up to that, we have learned last week. Now, last step, uh, when the two nucleus join together, how it looks like. So if this is the ovum, and what we can notice close to the ovum, so that is the zona pellucida. So and inside we can find a full set of chromosomes now. So it should be, it become now haploid. So we call it haploid now because half of the chromosome from the sperm and half of the chromosome from ovum fused to make full set of chromosomes in human 46. So we call it uh, not haploid, diploid. Diploid. Okay. Now the sperm cell, we can find the rest of the sperm cells. So this part of the sperm cell outside of the ovum. So then rest of the things, especially mitochondria, is not going, right? Sperm. So mid piece remain out of ovum. As a result, mitochondria from sperm not move into ovum. So mitochondria from sperm doesn't move into the ovum. So it's remain inside here, right? So this is now we call a zygote. This cell is called zygote. Zygote is a fertilized egg cell. So this is the last step. The first step is the releasing of the ovum. Second one is sperm cell and egg cell fused. At third stage, it makes zygote and the rest of the cells stay outside of the ovum. Okay. Can you also add this one at the last, at last step?
Okay, sir. Okay, sir.
Uh, so we have completed this section, fertilization of plants and mammals. In your workbook, what is the next lesson?
Yes. Particles 6 prepare and stay in a robotic course to observe the stages of mitosis.
A question or
It's page 35. Oh,
can you
it's
find a lesson called stem cell in 3C?
this and
3C
3C
stem cell.
stem cells. Yes.
Do you find stem cell?
Yes.
Okay. So we're going to talk about the stem cell. Now, why we want to talk about stem cell? If we look, what is the fate of this zygote? What is going to happen in this zygote? Before we start stem cell, uh, we can also learn what is the outcome of the zygote. So it is part of the stem cell. So we can learn stem cell by learning this fate of this zygote. Okay, now to understand that, uh, this is the oviduct or fallopian tube, and so this is connected to the ovary, right? So there's no direct connections. So this is the ovary, and this is the oviduct or fallopian tube. Right now, we can find the zygote after its fertilization somewhere here. Now, zygote is further moving because of the ciliary action of the oviduct or fallopian tube and muscular contraction of the oviduct or fallopian tube. The zygote is moving this direction. After that, it should reach uterus, and this is a uterine wall. Okay. Now, during this journey from the middle to the uterus, the cell zygote divide by mitosis. Okay. Zygote divide by mitosis. So we can find maybe two cell like this, and then after that it divide to make four cells. And then it is divided to make eight cells. 1, 2, 3, 4, 5, 6, 7, 8. Right? So here, 2 cell, 4 cell, 8 cells. Now this keep on dividing until it comes to certain cells that is roughly around 32 to 46 cells. right? So here, this one. So this contain many cells, roughly around 32 to 64 cells, around 64 cells. Let's say 64 cells. Okay, now this structure is called morula. It is called morula. Right? Now we can keep on going. What's happened to the next? So then another point we can find this one. The morula changes structure and it form a cell layer like this and like this. So right? Okay. Now inside there is a fluid filled space. So fluid filled space, this one, the middle, fluid filled space, which is called cyst. So that can also called cyst. So this one is called blastocyst. This one is called blastocyst. Now from blastocyst, this part, which is blue in color, this one make a placenta, this side make embryo. Now when it comes to a blastocyst stage, so this may be a 6 days after fertilization. 6 days after fertilization. Right now, we can talk about the stem cells. So if we further want to know what happened to blastocyst, uh, blastocyst slowly moving to the uterine wall, and then after that, uh, by about 8 to 9 days, so somewhere around 8 to 9 days, embed, 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 embed in uterine wall. That means when it comes to this place, it just uh, attached to the uterine wall, and then start to grow as embryo and grow as embryo. That is called implantation. So here it just arrive and embed in the uterine wall. So here 9 days the blastocyst further develop and embed in the uterine wall. That is called implantation. Okay. Now we can talk about stem cells. So once we understand this process, then only we can talk about stem cells, right? So the topic is stem cell. Can you copy this down under the stem cells?
Okay.
Right. Now, if we take cells of the embryo, right? So let's so this is the cells of the embryo, and for learning purpose, just try to draw a few cells. Okay. Seen from the previous diagram, zygote divide by mitosis. Zygote divide by mitosis. Now, if we go back to understand what is mitosis, we have learned mitosis previously. Now, if we try to recall what is mitosis, mitosis is nuclear division to form genetically identical cells. It's a form of nuclear division to make genetically identical cells. According to that, what we can expect, uh, consider this cell. So just consider this cell, and this cell can divide to make genetically identical two cells. So what will happen if it undergoes mitosis? So we can see like this. Two cells form. Now likewise, the cells are dividing, right? So, so here we can make one remark. Cells divide cells divide indefinitely. The cells are dividing indefinitely. Okay. Another thing is we also know that when the embryo develop, the embryo is developing different structures: head, hand, legs, uh, eyes, a heart, lung, different organs. So then we can also understand another process. That means these cells going to form the heart of the embryo. So if it is going to form heart of the embryo, so these cells are responsible to make the heart of the embryo. So it can form heart muscle cells. Right now, we understand two things. Cells divide indefinitely. These cells divide indefinitely, and some of the cells, they're going to move this direction to make the heart muscle cells. Right? So if we try to understand this, if it is heart muscle cells are like this, so this is the heart muscle cell. Now this cell changed to this. This cell changed to this. This is called cell differentiation. So during the embryonic development, certain cells divide indefinitely, and then some of the cells, right? So some of the cells, so this is one, and that's a two. Some of the cells change into different cells, right? Now, in stem cells, so stem cells, stem cells are un uh, they are undifferentiated. That can divide indefinitely. Stem cells are undifferentiated cells that can divide indifferentiated. And some of the cells can differentiate into another cell types. Now we can define stem cells. Stem cells are undifferentiated cells that can divide indefinitely. Some of the cells can differentiate into another cell type. So this is the definition of a stem cells. Now we can learn this from the embryo, because when embryo make like 8 cells, 16 and 32 and 64 cells, they are same. These cells doing two things. One is dividing, and the other one is some of the cells changing. dividing and changing. Some of the cells, these cells are dividing, and then some of the cells changing. So if a cell can do these two functions, they fall under stem cells. So stem cells come into this category, into this definition. They can divide and they can differentiate into different cells. Okay.